Namaste to all the students. Khushbu, myself, JV, Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about artificial chromosome, which is BAC. What first of all you have to understand by artificial chromosome vectors, and this session is related to recombinant DNA technology course of program BTech FPT 4th sem. And first of all, artificial chromosomes are what? So artificial chromosomes are DNA molecules that assembled in vitro from defined constituents which guarantee stable maintenance of large DNA fragments with the properties of natural chromosomes. Artificial chromosomes are useful for genome sequencing programs, for functional characterization of entire genomic regions, and for the transduction of large DNA segments into human and non-human mammalian cells. There are various types of the artificial chromosome vector, which is BAC, YAC, BAC, and HAC. If we talk about the cloning vectors and their insert capacity comparison, so if vector is plasmid, so host cells are E. coli and insert capacity is 0.1 to 10 kilobase pair. In the bacteriophage delta, bacteriophage gamma, host cell is E. coli and insert capacity is 10 to 20 kilobase pair. In plasmid and phosmid, host cell is E. coli and 35 to 45 kilo base pair capacity for insert DNA. And bacteriophage P1 vector, basically host cell is E. coli and 80 to 100 kilo base pair insert capacity. For BAC, means bacterial artificial chromosome, host cell is E. coli and 50 to 300 kilo base pair insert capacity. For P1 bacteriophage derived AC, means PAC, E. coli is host cell and 100 to 300 kilo base pair insert capacity. For YAC, host cell is yeast. Yeast artificial chromosome is the full form of YAC, and yeast will be host cell and 100 to 2000 kilo base pair insert capacity. For human AC, means human artificial chromosome vector, host cell is cultured human cells and insert capacity is more than 2000 kilobase pair. Now we will discuss about bacterial artificial chromosomes in detail. So a bacterial artificial chromosome, BAC, is a DNA construct based on a functional fertility plasmid, which is F plasmid, used for transforming and cloning in bacteria, usually E. coli. They are capable of carrying approximately up to 300 kilo base pair of inserted DNA sequence. What are the common gene components in BAC, bacterial artificial chromosome, which is RAP-E for plasmid replication E. So for plasmid replication and regulation of the copy number, PAR-A and PAR-B for partitioning F plasmid DNA to daughter cells during cell division and ensures stable maintenance of the BAC. Third one is the selectable marker, which is required for the antibiotic resistance. Some BAC also have leg Z region at the cloning site for blue-white colony selection. Next component is T7 and SP6 region. So phase promoters are required for the transcription of inserted genes. So these are the key points which are major components in the BAC: RAP-E, PAR-A, PAR-B, selectable marker. It can be leg Z or antibiotic resistant gene, and T7 and SP6. Here you can understand about the BAC structure in detail with the diagrammatic representation. So here I am sharing the slide of BAC vector. This is BAC vector P-BAC 108L, and it is having PAR B region, PAR A region. This is RAP-E region, origin of replication, And this is multiple cloning site which is having SAL1, SAC1. This is SAL1 and this is SAC1 region. This is cause and region. And loop locks P1, not 1, and hinge third, BAM H1. These are the P7 promoter and SP6 promoter. This is eczema, SMA1, not 1, BGL1 and SVI1 cloning restriction sites are present for the specific respective restriction enzymes. So this is the basic structure of bacterial artificial chromosome. Now we can discuss about the application of BAC vector. So our contribution to models of disease like inherited disease. So basically BAC vectors is able to cure the diseases, and BAC are now being utilized in modeling genetic diseases, often alongside transgenic mice. BAC have been useful in this field as complex gene may have several regulatory sequences upstream of the encoding sequence, including various promoter sequences that will govern a gene expression level. BAC have been used to study neurological diseases such as Alzheimer's disease, or, as in the case of aneuploidy associated with Down syndrome. There have also been instances when they have been used to study specific oncogenes associated with cancer. Now come to the contribution to models of diseases, especially infectious disease. So the genomes of several large DNA viruses and RNA viruses have been cloned as BAC vector, and these constructs are referred to as infectious clones. The infectious property of these BAC has made the study of many viruses such as herpes virus, pox virus, and coronavirus more accessible. If you talk about the sequencing, so BACs are often used to sequence the genomes of organisms in Genome Project. For example, the Human Genome Project. A short piece of the organism DNA is amplified as an insert in BAC and then sequenced. Finally, the sequenced part are rearranged in silico, resulting in genomic sequence of the organism. So this is the sum of BAC vector. Now we are going to introduce about artificial chromosome, which is YAC, yeast artificial chromosome. So yeast artificial chromosomes are genetically engineered chromosomes derived from the DNA of the yeast, Saccharomyces cerevisiae. Yeast artificial chromosome vectors allow the cloning within yeast cells of fragment of foreign genomic DNA that can approach 500 kilobase pair in the size. If you talk about the component of the YAC, so the vector contains several elements of the typical yeast chromosomes, including a yeast centromere, CAN4, yeast autonomously replicating sequence ARSI, yeast telomere TEL, genes for YAC selection in yeast, bacterial replicating origin, and bacterial selectable marker. Here you can understand the structure of YAC by diagrammatic representation. So here I am sharing the screen by which you can understand more about the YAC vector. Here is the example of YAC vector, which is P by AC4, which is 11.4 kilobase here. Here you can simply see this is the Hind3 exo1 cutting region. This is TEL region, which is yeast telomere region. This is TL region, and in between them BAM H1 cutting region, Hind3 exo1 cutting region. This is origin of replication, ampicillin resistant gene and TRP1 region. URA third region, this is also cutting region, and ARS1 region, which is known as autonomously replicating sequence, and CEN4 region means yeast centromere region. So these are the specific points present in yeast artificial chromosome. If you talk about the construction of YAC, so a YAC is built using an initial circular DNA plasmid, which is typically cut into a linear DNA molecule using restriction enzymes. DNA ligase is then used to ligate a DNA sequence or gene of interest into the linearized DNA, forming a single large circular piece of the DNA. Here you can understand it by diagrammatic representation again. You can simply see this is the origin of replication. This is centromere region, eco-R1 cutting region, selectable marker A, selectable marker B. This is telomere region, and this one is the BAM-H1 cutting region. So first of all, digestion with the BAM-H1. So it will remove this section. This section will be removed, and the remaining portion will become linearized. These are the telomere regions. And now this linearized region will be digested with the eco-R1. So eco-R1 are basically this region. So it will eco-R1 will cut the, this is the eco-R1 region. So it will cut the region, and that's how small region will cut, and this is the large region will be cut. And this region is having selectable marker B. This region is having only selectable marker B. Both are having telomere region, and large area is having centromere region and origin of the replication. Now, fragments of the genomic DNA collected, and they are bound by the high digestion with, they are also treated with the eco-R1 and further ligate. So these foreign DNA fragments will be introduced in between this region. And this recombinant YAC vector, linear YAC vector, further transform into the yeast spiroplast cells. Further, you can go for the selection of yeast cell is recombinant or not. So you can go for the selection, or you can go for the enzymatic digestion of the cell wall. So that's how recombination occurs in of YAC plasmid, YAC vector into the yeast cell. If you talk about the applications of the YAC, so yeast expression vectors such as YAC, yeast integrating plasmids, and YAP, yeast episomal plasmids, have an advantage over bacterial artificial chromosome in that they can be used to express eukaryotic proteins that require post-translation modification. By being able to insert large fragments of the DNA, YAC can be utilized to clone and assemble the entire genome of an organism. With the insertion of YAC into yeast cell, they can be propagated as linear artificial chromosome, cloning the inserted regions of DNA in the process. With this completed, two processes can be used to obtain the sequenced genome or region of insert. First one is the physical mapping, and second one is the chromosome walking. What are the limitations of the YAC vector? So very large DNA molecules are very fragile and prone to breakage, leading to the problem of the rearrangement. High rate of the loss of the entire YAC during mitotic growth. Difficult to separate YAC from the other host chromosomes because of their similar size. Separation requires sophisticated pulse-filled gel electrophoresis, which is PFGE, and yield of DNA is not high when the YAC is isolated from yeast cells. Clones tend to be unstable, with their foreign DNA inserts often being deleted. So this is the sum up of artificial chromosome vectors. And this session is powered by digital version 2.0 JAYOTI Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.